Okay, good afternoon, everyone, and thank you all for hanging in there and sticking it out to the end. I'd like to just thank the organizers for inviting me to speak today, and those of you that are probably wondering what does cancer and disparities have to do with climate change and all these other topics we've been talking about. I've just put some of the UN, the Sustainable Development Goals that this particular project I'm working on, I think, is most relevant to. So I wanted to start by just giving a little story about myself and how I got to be doing what I'm doing. I was born and raised in the beautiful island of Barbados in the Caribbean. I came from a low-income family, but my parents and my neighbors and everyone recognized I was a unicorn and quite different. And so they saved as much as they could to send me to university for the first year. And I chose Queen's University in Canada because they acknowledged me as an individual before I even got there, because this was pre-internet, Google, everything, and we couldn't afford to go see the universities. But they were the only university I applied to that actually sent me information every other week before I had to make a final decision as to which university to attend. So I went to Queen's, had a marvelous time. However, in the last year of my, in the last year of my undergrad degree, my neighbor in Barbados passed away from breast cancer. My mother was diagnosed with ovarian cancer, and then she succumbed to ovarian cancer 6 months later and 5 days before I convocated from Queen's. So at that point, I was heading towards medical school, and I decided to pursue cancer research instead, because I felt that the doctors were not able to save her because we did not have enough drugs and therapies for ovarian cancer. And sadly, we still don't have enough drugs or therapies for aggressive cancers like ovarian cancer or pancreatic cancer. At that point, the novelty of Canada and the cold weather had worn off, and I chose to do a postdoctoral fellowship in Memphis, Tennessee, because I thought it was a lot further south and would be warmer, like the Caribbean. And I chose to work in Dr. Al Reynolds' lab because during my graduate studies at University of British Columbia, I realized that the one hallmark of cancer that really led to the demise of most patients was the fact that the tumors broke away from their primary tumor site and spread to other organs such as the lungs, the liver, and the brain. So Dr. Reynolds at the time had just discovered a new gene himself, and it was one of the first substrates that was identified for one of the first oncogenes that was identified in the mid-80s that contributed to human cancers. But no one knew how the SRC kinase actually caused cancer. And Al was part of the team in Virginia that identified many of the SRC substrates that people around the world are now studying. And so I chose to work in Al's lab because he was the only person studying P120, and I felt that anything I did in his lab would actually have some impact. So he was studying this protein. Where's the pointer? Yeah. So he was studying this protein P120 that he identified as associating with the transmembrane cell adhesion protein, E-cadherin, and 50% of all human metastatic tumors are due to malfunction of that E-cadherin-catenin complex. So my role in his lab, or my project, was to determine the role of this new protein that he had identified to understand how it was causing metastasis in human tumors. So during the process of characterizing P120, I cloned a novel gene, just like he did, that was not characterized. And he gave me permission to name it anything I wanted. Being from the Caribbean, not seeing many black women, far less black scientists, I decided to name it something that would be a little, Caribbean. And so I named it Kaiso after my favorite calypso music. And this is just a photo of the dancers in the Caribbean and in Toronto when we have our annual Caribbean Festival. And over the first 8 years or so as a faculty, why are the slides all kind of weird? Okay. It's not going back. Okay, we'll just leave it there. Okay, so the first few years as an independent professor, I spent my time characterizing Kaiso, determining what it did, and determined that it was a dual specificity transcription factor, meaning that unlike most transcription factors, it bound to 2 different types of DNA, which meant that it regulated many more genes than a typical transcription factor did, which meant it would also be harder to really characterize in depth, but it meant that it was not a low-hanging fruit. We also found that it was implicated in many biological processes. Most of these were all processes that contributed to cancer: cell proliferation, cell motility, inflammation, etc. And my lab and others found that it played a role in multiple cancers. So while I was doing this, I kept thinking, this is not why I pursued a PhD. I really wanted to do research that would have global impact. That was my childhood dream growing up in Barbados. And I felt like I was doing fundamental research, which was great, but I wasn't seeing that human application. And then in 2008, I attended the American Association for Cancer Research Conference and heard the physician there, Dr. Lisa Newman, talk about a breast cancer subtype that was disproportionately affecting Black women in America as well as in West Africa. And that really piqued my interest because I had never heard about this, even though I'd been doing cancer research for over a decade. And at McMaster, they were also encouraging us to think about retirement/refreshment. So I was already thinking about what can I do to give back to the Caribbean with my cancer research efforts. Oh, the slides just they were fine earlier. Anyway, so breast cancers are classified based on the expression of 3 biomarkers that some of you, hopefully, or all of you have heard about: the estrogen receptor, the progesterone receptor, and the HER2 receptor, which should have been here, but you can't see them. But basically, this is the cell, these are the 3 receptors. And the subtype of breast cancer, triple-negative breast cancer, those cancers or tumors do not express any of those biomarkers. And the reason that this is problematic is because the current treatments for breast cancer specifically target estrogen receptor or the HER2 receptor. So we have tamoxifen that targets estrogen receptor or ER, and Herceptin that targets the HER2. Triple-negative breast cancer patients, however, do not express any of these biomarkers, consequently cannot be treated with these drugs, and can only be treated with standard chemotherapy or radiation therapy. And as a consequence of this, they have the highest mortality rate of all diagnosed breast cancers. As I said, when I first heard about this, heard Lisa Newman talk about this was really disturbing, one, being a Black woman, and coming from the Caribbean, where I know that we were ancestrally linked to women in West Africa, where she was seeing an incidence of triple-negative breast cancer as high as 50% in the women that were going to the hospitals there. So what is triple-negative breast cancer? I've just told you. So it's highly prevalent in young premenopausal Black women, as well as some young premenopausal Hispanic women. There's a poor prognosis, which we know is due to the lack of these specific therapies because they don't express ER or HER2. And initially, when I first heard Dr. Newman talk about this and when I read the literature, most of the studies were suggesting or implying that this poor mortality and high prevalence was due simply to the low socioeconomic status of Black women in West Africa and in the United States. So while socioeconomic status might explain the outcomes and poor mortality or high mortality rates, it wouldn't necessarily explain the high incidence in these populations. Notably, the World Health Organization has indicated that breast cancer is actually an emerging health crisis in Africa, since the 5-year survival from breast cancer in Africa, in sub-Saharan Africa, is less than 50%, compared to 90% in North America and European countries. So this is really problematic. Sadly, the US is one of the only places that collects race-based data. So most of our studies and the studies of those of us studying triple-negative breast cancer are based on data collected in the US, because in Canada, for example, we do not collect race-based data. We've just started to collect race-based data, and it will take at least 5 to 10 years before we have enough data to analyze in the Canadian context. But I wanted to show this slide to show you that, historically, NHW refers to non-Hispanic white or white women, and NHB is non-Hispanic Black women. So historically, white women have had the highest incidence of breast cancer since they started collecting the data. This graph doesn't show it, but they've been collecting this data since 1975 in the US. And it has always been predominantly white women with the highest incidence of breast cancer. And hopefully, and on this graph here, which is now showing the trends in mortality, you can see that there's suddenly this switch from white women being at the top of the graph. You see this sudden decline in the mortality rates for white women, but the mortality rates for Black women kept increasing slightly and then started to decrease, but they remained the highest mortality rate of all the ethnic groups in the US and significantly higher than all of these other groups. We believe, or I think we're pretty confident, that this decline that we see here in white women was due to the introduction and the development of tamoxifen and Herceptin, which most white women tend to express those biomarkers. They tend to have ER-positive tumors or HER2-positive tumors. And sadly, most of the clinical trials that were done when these drugs were being developed were done predominantly on white women and not diverse populations. And because if they had been, they might have noticed that the Black women were not responding to those drugs. Next slide. So as I said, I first heard Dr. Newman in 2008. My next research lead was scheduled for 2011. So in 2011, I went to Barbados and started collaborating with physicians there, as well as in Nigeria. And this is just a diagram of a schematic of the globe showing you the incidence of triple-negative breast cancer based in, West Africa? Yep. So in West Africa, the incidence of triple-negative ranges from 50 to 60%. This 15% is predominantly in East Africa, which is not where most of the transatlantic slaves originated from. So we believe that the high incidence in the Caribbean and in Black American women is due to being descendants of these ancestors in West Africa. So as I said, I started collaborating with oncologists and researchers and pathologists in the Caribbean as well as in Nigeria. And we collected triple-negative breast cancer samples from these women. And then we did this high-throughput study called tissue microarray analysis, where we put small pieces of tissues from each of these patients onto one slide, which allowed us to analyze the expression of different genes and biomarkers simultaneously on multiple patients, rather than doing it one at a time, which was historically how it was done in the past. And in the interest of time, I just wanted to show you the results that we found when we looked at the expression of the gene Kaizo that I had cloned and discovered. And the reason we looked at this was I had collaborated with a scientist in the Netherlands, and we had found that Dutch women with very aggressive invasive ductal carcinomas had significantly higher levels of Kaizo expression compared to Dutch women with non-invasive type cancers. And in this, I hope you can appreciate and see in this slide that the Nigerian and Barbadian women had significantly more Kaizo expression, as indicated by the dark brown staining in the nucleus and the cytoplasm of the cells, compared to African American women and compared to Caucasian women. Oh goodness, I can't believe these slides they were fine. Anyway, so alongside while we were doing this, we also looked at the clinical pathological variables of women in Barbados diagnosed with triple-negative breast cancer. And here, unfortunately, you can't see it. My gosh. Am I supposed to be pointing at this, perhaps? Maybe. But the red rectangle there was highlighting the fact that triple-negative breast cancer patients in Barbados had the highest grade tumors. As I said, because we were noticing this high level of Kaizo expression, we then generated some Kaizo-depleted cell lines to see whether that would cause morphological changes in the cells, which they did. And you can see here, these are the parental, highly metastatic cells expressing lots of Kaizo, very little of that protein E-cadherin that mediates cell-cell adhesion. When we deplete or remove Kaizo, we get the restoration of E-cadherin, and the cells become epithelial again, which means they're less motile. We then confirmed this using mouse models where we put these Kaizo-depleted as well as the parental cells into nude mice, into the breast mammary fat pads of the mice, and then looked to see whether the removal of Kaizo would prevent the metastasis or spread of these triple-negative breast cancer cells to the lungs and the liver. And we were pleasantly surprised and excited to find that indeed the simple removal of Kaizo prevented the metastasis of these tumors. As you can see here, the parental cells had lots of metastasis in the lungs and the liver, indicated by these darker pink regions outlined in white, whereas the mice that had the Kaizo-depleted triple-negative cells hardly had any metastasis. So these are just some of our ongoing and future studies. We're trying to determine the cause of high Kaizo expression in aggressive breast cancers. We're developing prognostic and diagnostic kits to see if we can look at Kaizo expression to help physicians and women determine whether or not the types of therapies and perhaps surgeries that they should consider. And we're characterizing some of the genomic profiles of women in the Caribbean. And just to wrap up, I just wanted to mention, in case you didn't realize or know, that we're wrapping up the end of the International Decade for People of African Ancestry or Descent, which represents about 1.5 billion people globally and 18% of the global population. And again, because most of this work is being done in the US, this is a statement from the American Association for Cancer Research in their annual Cancer Disparities report. This is from 2020, where they've added racism trauma as a social determinant of health. And they've added this to determinants such as education, income, etc. So a few years ago, in the theme of the Human Frontiers, Canada has adopted this approach, and we now have a New Frontiers program, funding program in Canada, and I was fortunate enough to be funded for this program along with two collaborators, Dr. Ingrid Waldron, who is a social scientist, and Paula Marignani, who is another molecular biologist like myself, and we're doing this in partnership with a community member from this small community in Nova Scotia, called Shelburne. And the Shelburne community is a community that was established 400 plus years ago by slaves that were brought over from West Africa, as well as some were brought from the Caribbean. And this small community has an incredibly high and unusually high incidence of multiple different types of cancers. And they believe that it could be due to toxic chemicals that are in their community, since a dump, unfortunately, a biohazard dump was put in that community 75 years ago. And so we're doing the study to try to see whether or not that is a contributing factor to the high incidence of cancer, or whether it's due to perhaps some ancestral mutations that they have. And this is just some recommendations in general. If we want to improve the health of the planet, we need to definitely address and improve Black women's health in particular, because we're suffering, and we want to contribute to society. And so we need increased representation of Black healthcare professionals and researchers. There's not enough of us, definitely not in Canada. And I think this just me and some of my trainees. And just to say that if we do this and we have some representation with agency, I heard everyone talking about more inclusive stakeholders and diversity. And so I'm just, as I said, I'm just trying to showcase that if we can tackle this problem together, we will have this beautiful world, hopefully, that we can continue to live on for longer than we anticipate at the moment. And I'll end there. Thank you.